here. And title is INFIMA leverages multi-omic model organism data to identify target genes for human GWAS variants. And our main objective is to do fine mapping of DO mice islet EQTLs in order to deorphanize human GWAS SNPs. So what is DO mice? DO mice is short for diversity outbred mice. And it starts from the 8 founder mice and contains about 60 million SNPs. And then they outcross and we obtain DO mice, which resembles the genetic diversity in humans. The motivation of the study originates from the DO EQTL analysis. And LOD score is actually a measure of evidence of strength of the presence of EQTL at some locus. And this example is that we identify a strong EQTL for this capsule gene, and the corresponding allele effects is in the bottom. But if we zoom in this locus, we can see a lot of mouse SNPs that have high LOD scores, and we have no way to discriminate the true causal SNP, and this is the limitation of EQTL analysis. Now, so what to do? We want to fine map by integrating DO EQTL data with additional functional genomic data. The first piece of data we have is the attack from founder mice. We have 8 founder mice, 16 samples in total. And in general, genes with higher expressions tend to have more open chromatin in their promoter regions, but that's not always true. But for this example, capsule gene, we identify a differential attack peak at its promoter region, and is consistent with the allele dependence from the DO EQTL analysis. So what drives the differential signal of the attack peak? We would like to find the SNPs covered by those attack peaks. Maybe those SNPs affects motifs and changes TF bindings, then therefore affects the chromatin accessibility. And we first try to get the SNPs covered by the attack QTL and find the SNPs that strongly correlated with the attack peak, that is called local attack QTLs. And for these 47,000 local attack QTLs, we perform motif analysis as well as footprint analysis to find evidence of transcription factor change caused by these SNPs. Here's an example still at the promoter locus of this capsule gene. We identify a SNP, mouse SNP, that contains alternative allele T for PWK and Cas strain that disrupts this PAX motif, and which is consistent with one literature saying that PAX TFs regulate human capsule gene. Another piece of data we have is the founder mice RNA-seq data. And we have 91 samples and more than 13,000 protein coding genes. And we observe very beautiful consistency between the founder gene expression with allele effects from the DO EQTL analysis. Now our question becomes, how do we leverage this data to address the allele dependence we observed? And we developed a model called INFIMA. INFIMA is short for integrative fine mapping with model organism multi-omics data. The idea is pretty straightforward. For each DO EQTL gene, say we have DO EQTL marker in the middle, which located in the gene exonic region of gene 1. And our method is to search around 1 megabase radius, which is typical threshold for cis-EQTL analysis. And we identify a lot of attack peaks within this window, and the local attack QTLs covered by those attack peaks are treated as candidates for this DO EQTL marker. So which one is causal? Maybe the one closest to the gene 1 promoter or maybe the one that is closest to the marker. Our model resolved this issue. This model is a latent factor model, and we fit the model with expectation maximization technique. Model quantifies how well each non-coding SNP explains the observed DO allelic pattern through the consistency of each piece of data we mentioned before. The founder RNA-seq data, founder attack-seq data, which contains the existence and consistency of a footprint. Also, the observed allelic pattern, the DOE-QTL data. The output we get from this model is the SNP level quantification from the model, indicating, essentially estimating the posterior probability for each local attack QTL, the causal posterior probability of that DOE-QTL. And we validate our model, use the high-C data from mouse islet. For a given gene, we evaluate the high-C signal between the causal local attack QTL and the promoter of the target gene compared to several baseline methods. And here is one figure of cumulative empirical probability distribution validation technique. And so the dashed lines are baseline methods. Maybe we just randomly pick one causal SNP, or that SNP closest to the marker or the closest to gene. And the solid blue lines are the best causal SNPs predicted by our model. And the yellow line is the worst model, and even worse than the random line. And our the best results, as expected, is the blue solid line. One interesting observation is that randomly chosen one causal SNP is worse than the SNP closer to marker and worse than closer to gene. Now, our goal becomes, how do we use these fine mapped DOE-QTLs to link to GWAS SNPs, or help us to understand the GWAS SNP and the gene linkage. We here use synthetic mapping and validated by promoter capture high-C data. And this is a simplified version of mapping scheme. We have more than 14,000 GWAS SNPs of 16 complex human traits associated with pancreatic islets functionalities, like type 1 diabetes, type 2 diabetes, blood glucose, and so on. So we first use leftover tools to map from, so here we have human attack data and mouse attack data. And we try to map the human attack peaks with mouse attack peaks using leftover. And the human attack peaks can cover GWAS SNPs, or the GWAS SNP is very close to that peak. And here, the mouse attack peak can cover several SNPs that is correlated with the attack signal. And these local attack QTLs predicted by INFIMA model can be linked to gene 1. And so say like gene is linked to mouse attack peak. And this gene is not necessarily the nearest gene. And we compare our method with the linking attack peak to their nearest gene. And then we link mouse attack peak to mouse gene. And we find the orthologous gene in human, and to check if there is a PC high C signal between the human peak and the gene promoter region. And then we complete our mapping here. As mentioned before, we validate these predictions and compare with linking the attack peak with their nearest genes. And we found enrichment results. The results predicted by INFIMA model is enriched with this promoter capture high C data. Here is one example of our mapping. This is an NAA60 example in the mouse genome. And in mouse gene NAA60, we is EQTL markers far away. It's around 1 megabase. And the INFIMA model predicts that this mouse SNP is causal for this DOE-QTL. And this SNP is not necessarily the closest SNP to this gene. So you can see a SNP here. And for the mapping, the mouse SNP maps to one GWAS SNP for body mass index trait. Then we move to human scale. In human, we can see the NA60G gene is here and the promoter region upstream 3K. And then we see a beautiful promoter capture high C link the promoter region to that body mass index gene mapped to mouse SNP. And it's covered by one human attack peak as well. In summary, we developed an INFIMA model for integrative fine mapping of DO mice EQTL, and we are able to link GWAS SNP to genes with synthetic mapping. Finally, I would like to thank my advisors, Sundus Kalish, and all of my group members and the amazing collaborators. And thanks for your patience.